The first step in DNA purification is to break open the cells from which the DNA will be obtained. This is simply achieved by lysing the cell with anionic detergent such as sodium dialysyl sulfate SDS, or in some cases such as in plant cell or in bacterial cells. To rupture the cell wall, we have to use some enzymes such as lysozyme, and to some extent we could also use EDTA, which is a chelator that can chelate calcium or magnesium or divalent cations. Once the cells have been broken, two different approaches can be used to purify the DNA from the resulting extract. The first involves degrading or removing all the cellular components other than the DNA, a method that works best if the cell do not contain large amount of lipid or carbohydrate. The extract is first centrifuged at low speed to remove debris such as pieces of cell wall, which could be collected at the bottom of the tube. The supernatant is then transferred to another test tube and mixed well with phenol, which causes the protein to precipitate at the interface between the organic and aqueous phase. The aqueous layer, which contains the dissolved nucleic acid, is collected and a ribonuclease enzyme added into it. Acid is so a ribonuclease enzyme is added, which breaks the RNA into a mixture of nucleotides and short oligonucleotides. The DNA polynucleotides, which remain intact, can now be precipitated by adding ethanol. In the second approach to DNA purification, rather than degrading everything other than DNA, the DNA itself is selectively removed from the extract. One way of doing this is by ion exchange chromatography, which separates molecules according to how tightly they bind to an electrically charged particle in a chromatographic resin. DNA and RNA are both negatively charged, as are some protein, and so binds to a positively charged resin. The binding, which involves ionic interactions, can be disrupted by adding a salt solution with the less tightly bound molecules detaching from the resin at relatively low salt concentrations. This means that if a solution of gradually increasing salt concentration is passed through that column, then different types of molecule will lead in a sequence from protein to RNA to DNA, reflecting their relative binding strengths. In fact, in a very straight way, 1 molar NaCl at pH 7 is sufficient to elute the protein and RNA, leaving just the DNA behind, which can be, and the DNA can be eluted very easily by using 1.25 molar NaCl at pH 8.5.